Hello, everyone. I'm Simon Heeke. I'm an assistant professor at the MD Anderson Cancer Center in Houston, Texas, where I'm also leading the liquid biopsy translation working group. And therefore, I'm very excited to give to you today an introduction to liquid biopsies for precision oncology. And in this talk, I will really focus on the treatment selection and the monitoring in later stages of the cancer care. Here's my disclosure. To directly start to give you an introduction to liquid biopsies, I'm very sure that many of you have already heard a lot about liquid biopsies, but just to refresh everybody's mind, in the same way as a biopsy is a way to access a tissue material, for example, from a cancer, but also from different organs, to gather more insight on the biological processes or for treatment selection, for diagnostics, the same way a liquid biopsy tries to achieve the same using very different body fluids. The idea is practically the same, that, for example, when you have a cancer or you have another disease, that material from this cancer is spread into this body fluid, and thereby by analyzing of it, we really get access and insight into the biological processes of the organ or the disease we are interested in. The important thing today, we're speaking about liquid biopsies in the context of cancer, and especially in the context of cancer in the late stages. But it's important to highlight that while it's very frequently used in cancer, liquid biopsy isn't something that is only limited to cancer, but is also developed in other disease entities. Importantly, today, when we think about the way we are using it, it is commonly used to either replace, but rather to complement other methods that are occasionally more invasive, such as, for example, acquiring a tissue biopsy to really get insights into the tumor intrinsic processes for treatment selection, for biological research, for translational research, but really to understand the processes within the cancer. The important thing is liquid biopsy is not something of the future, but actually liquid biopsies are already approved by the FDA and other regulatory organs for some indications. And therefore, liquid biopsy already today play a very important place, for example, cancer diagnostics. When we now think about, and as I've mentioned, liquid biopsies really allow us to get insight into processes that are happening within the cancer. And this is really because we know that cancer cells are capable of releasing some biological material into, for example, the bloodstream that we can then use, analyze, and therefore make our decisions. And in fact, there are two processes that are happening, all of them in parallel. And one is really a passive release of those biological materials, for example, because cells are dying via apoptotic or necrotic processes, for example. But there is also an active release, for example, by the spreading of extracellular vesicles and other tumor-derived material. And this is really important because this really means that we have a huge plethora of different tumor-derived material within, for example, in this case, the bloodstream, that we can really analyze to gain insights into the processes within the tumor. While there are many other processes, circulating tumor cells, for example, or extracellular vesicles, I really will try to focus my presentation today on two very important entities, which is circulating tumor DNA and circulating tumor RNA. The important thing there is that while both nucleic acids are really spread into the bloodstream by all those processes, the idea is that commonly circulating tumor DNA is a little bit more derived from passive release, while we see circulating tumor RNA much more spread into the bloodstream by active release, for example, by being bound into extracellular vesicles. The important thing is both ctDNA and ctRNA now allow us to really get more insights into processes that are happening within the tumor that we can then use to either get biological insights or we can use it for diagnostic purposes. When we now think about circulating tumor DNA, which is the much more used entity for liquid biopsies right now, we can really use circulating tumor DNA in pretty much the same way that we are using tumor-derived DNA for diagnostic purposes. We know that we can use circulating tumor DNA, for example, to detect specific mutations, for example, in the EGFR gene that we can use for treatment selection. We can use circulating tumor DNA for copy number alterations, for example, amplifications in the HER2 gene, for example. But we can also use circulating tumor DNA to derive much more interesting signals, for example, from epigenetic modifications. So for example, DNA methylation, since this signal is really preserved into circulating tumor DNA. Or we can use circulating tumor DNA to also detect larger chromosomal rearrangements, like gene rearrangement, for example, like ALK gene fusions, but also much larger chromosomal rearrangements. In addition to the detection of circulating, to the detection of those modalities using circulating tumor DNA, we can also use circulating tumor RNA to actually get more insights into the cancer. And there are two big themes which I really want to highlight today. And one is simply, as we know that we use RNA as a method to detect gene expression, and this is really something that we can also use from circulating tumor RNA, where we can really try to derive much more important information on whether genes are expressed or not expressed, whether genes are differentially expressed between certain groups. But very interestingly, and I will explain this much more, is that we can use circulating tumor RNA also for the detection of gene rearrangement, and potentially can actually use it for better detection of gene rearrangements. And the idea behind is that when we now think about gene rearrangements, again, many of them where we have really therapies associated with it, then what we are actually having is that we always have two different genes. Let's call them gene A and gene B. And when we now look at the DNA, I think we all know that each gene consists of exonic regions, of intronic regions. And then there are genomic events that lead to the creation of a fusion DNA, where we now have gene A or parts of gene A fused to parts of gene B, which is then creating this new fusion transcript. When we now looked at from the circulating tumor DNA, then what we will actually see is a strand of DNA with a couple of exonic regions, with intronic regions. And when we now try to sequence this, we really have to see if we can actually find the position where we have the overlap of those two genes. But when we now look at the RNA transcript, which is practically our DNA transcript, but after splicing, we have all those intronic regions removed from it, then what we can see that we only have those exonic regions stick together to create this fusion transcript, which means that when we now use this RNA-based analysis, we might have a chance to actually better find the breakpoint and to better identify our not only the presence of our fusion gene, but also where and how the fusion gene have been created. And this is a principle that is already used, for example, in tissue-derived analysis. What you can see here, for example, is an analysis that we did really with an integrated sequencer to really understand if we can get very quick turnaround insights into the presence of gene fusions. And we really, we're using two methods there. We were using a qPCR-based assay and an integrated NGS assay. And we're really looking across a span of different gene fusions that are relevant in lung cancer. And what you can actually see is with a method that integrates both DNA and RNA, this time from tumor tissue, not from liquid biopsies, we were really able to detect gene fusion with a very high probability. In fact, when you look at this, we were really able to achieve an extraordinary accuracy for the detection of genes, with only the ALK gene being a little bit more challenging with a more limited sensitivity. We've seen that there are other methodologies. I will not go too much into this today, like imbalance detection might actually help to boost a little bit the detection. But generally, I think this really emphasizes that the combination of DNA and RNA from tumor tissue is really excellent to detect those very important gene rearrangements. However, the question is, when we can now use it from tissue, how can we actually use this also from liquid biopsies? And in fact, it seems like that RNA-based detection for fusion detection is really helping to boost a little bit the detection rate. For example, this was an analysis that we did a couple of years ago. At that time, using a qPCR-based assay, we were really trying to detect ALK rearrangements again. And we're really trying to detect them using only circulating free RNA using a prototype qPCR-based assay. And what was very interesting when we look at samples derived from patients at baseline, during treatment, or at progression, we were really capable of detecting ALK fusions just from the analysis of circulating free RNA. But we didn't detect any of those fusions in patients where we know that there were no rearrangement present based on tissue analysis. So while this was very, very promising and really demonstrated that circulating free RNA can be used for the detection of gene rearrangements, we were really not that happy with the sensitivity that we had there. So it really seems like that this is a promising approach, but we really need to boost sensitivity. So what we did then, and what we published relatively recently, earlier this year, is that we now used an NGS assay, which combined actually both, which combined DNA and RNA. And using such an assay, we were analyzing samples from the Bright Star trial. The Bright Star trial was a trial analyzing brigatinib plus local consolidative therapy in patients with ALK rearranged non-small cell lung cancer. The important information is that those were patients with a known ALK gene rearrangements. And we were really looking at ALK gene fusions across different time points, for example, at baseline, prior to local consolidative therapy, after local consolidative therapy, and at progression. But really the focus here today is really to look at the detection of ALK gene fusions at baseline. Because when we now split the detection based on DNA and RNA-based analysis, what we could actually detect is that there were many gene fusions that we detected with both, with DNA and our RNA-based method. But on the other side, we could also see that there were certain gene fusions that were only detected by DNA or were only detected by circulating free RNA. And this is very, very important because this really highlights, together with the data that I showed you before using only RNA, that it seems like the best approach is really to combine both. Because we know that there are some gene fusions that we only detect in RNA, and there are some gene fusions that we only detect in DNA. And we really need to better understand why we see those differences there. The important information additionally is there that when we just look at the baseline ALK detection, we could demonstrate that this is really also associated with clinical outcome. So we don't only have the opportunity with liquid biopsies to detect and stratify a gene rearrangement and stratify patients accordingly, but to also get much, much more prognostic insights using this methodology. Here we were working with an outside provided assay, and we were really, really excited about the results. But I think the idea of combining circulating tumor DNA and circulating tumor RNA is something that is generally very, very feasible. That, whenever there's an opportunity within an assay to add another modality that might improve the detection of gene rearrangement, that I'm very optimistic that actually this approach is much more generally true. However, outside of the detection for treatment stratification, we can also use circulating nucleic acids like circulating tumor DNA or liquid biopsies in general for treatment monitoring. I think this is something that we have heard much, much more in the earlier stages where, for example, minimal residual disease is a term that is quite often used to define patients, which have tumor tissue left after, for example, curative therapy. But the important information is that treatment monitoring is also something that we can use in a metastatic setting. And it's really something that can help us to use liquid biopsy throughout the real course of treatment, from diagnosis to the treatment monitoring. We can use it to detect resistance to therapy and even continue to monitor it on the second line treatment. Because with the liquid biopsy, when we look at blood, for example, we really have the opportunity to do longitudinal tracking. And what we can then, for example, see is what I'm trying to show you here is, for example, there might be two different lines that we see. So we see a blue line and we see a green line. So on the green line, let's assume this is an important cancer mutation that we see there, and this is the circulating tumor DNA we detect. You see that this even drops below the level of detection. And now there are a lot of studies, for example, ongoing in very different settings, like can this, for example, be used as a biomarker to change the treatment, for example, to de-intensify the treatment? Might there be other treatment strategy that we can use when we are no longer able to detect circulating tumor DNA in these patients? All of this is currently researched. But we also see the blue line, for example, where we see that the detection of circulating tumor DNA upon treatment is not going below the level of detection, suggesting that probably there's more tumor tissue left, and maybe there might be alternative treatment strategy in such a case, which are very important. We can see that, for example, when we look at clinical progression, that maybe we might see an increase in the total level of circulating tumor DNA, or we might see the emergence of resistance mutations that might now tell us that actually the primary cancer has become resistant to the therapy, and thus would allow us to change the treatment. Again, this is just an example. This is not, as a recommendation for treatment, and there's a lot more research necessary to really understand the best way of using liquid biopsy. But I think it's very important to highlight in this context that even in a metastatic setting, we can actually get a lot more insight from liquid biopsies. And to give you one example, for example, of treatment monitoring, I would like to introduce to you the Ramosi trial. The Ramosi trial was a multi-center trial where we were testing the combination of osimertinib with or without ramucirumab in TKI-naive EGFR mutant non-small cell lung cancer. And the important thing is the Ramosi trial, which was published earlier last year, really highlighted significant improvement in progression-free survival when osimertinib is combined with ramucirumab, making this a very, very interesting clinical data that we have there. But now we really wanted to see how can we actually use liquid biopsies to better understand the dynamics and biological insights during the Ramosi treatment. And therefore, what we were actually doing is that we really looked at liquid biopsies at baseline, at cycle 3, day 1, as well as at progression to therapy. And we were really interested to specifically look at the amount or the variant allele frequency of EGFR mutant in circulating tumor DNA. Because the question that we had is a higher amount of detectable EGFR mutations in circulating tumor DNA, for example, prognostic of outcome to therapy? And while we will show much more clinical data later this year, and I'm very, very excited to provide more clinical insights about the clinical trial, I already would like to share with you, for example, that when we just look at the baseline detection of EGFR, we could actually see that we have a very excellent capabilities of detecting EGFR mutations. And this is irrespective of the two treatment arm, which is very important that we see a balanced detection. But even in patients who have brain metastasis, which is often associated to a much more challenging detection of EGFR mutations with liquid biopsy, we actually had an excellent and comparable detection of the primary EGFR mutation in liquid biopsies, highlighting that this is really a methodology that we can use to track those very specific mutations that are associated with the therapy. And when we now think about the way that we want to look, for example, at EGFR mutation, I think it's also very, very important to highlight, for example, that detection in the liquid biopsies of a mutation is surprisingly well correlated across certain assays. For example, here I'm going to show you some data that was really looking at the comparison of variant allele frequencies, so the fraction of a certain mutation, this time specifically looking at ASCAT1 or 2 mutations across 4 different liquid biopsy panels. This is a study that was led by Paul Hoffman in Nice, and I'm very grateful that he allowed me today to show some of the data with you. Because what I found really, really interesting is when we now look at the correlation of the variant allele frequency across those 2 assays, across different assays, we could really see that the correlation is absolutely excellent. And it doesn't matter if it's an amplicon-based assay, is it a hybrid capture-based assay, if we use an integrated sequencer, or if we use a classical sequencer, we could really see that the detection of a gene mutation from liquid biopsy seems to be really well correlated across assay, meaning that when you find something in a liquid biopsy, this is really an information that you can use. But now I spoke a lot about the use of liquid biopsies, how we can use it, how we can use the prognostic assay, how we can use it and compare across different liquid biopsy assay. I think it's also important to very briefly also show to you the comparison of liquid biopsy to tissue biopsy. And I don't want to share with you some information about sensitivity, specificity. There's a lot of research out there, and when you're interested in this, you will find a lot of very excellent papers who have actually done it. But I really want to speak about liquid versus tissue biopsy as a process of generating data. Because when we look at the tissue biopsy, obviously, we need to have a tissue. And this means that we have to make an appointment for a biopsy. We have to actually perform the actual biopsy. Then we have to fix and embed the tissue and finally do the next generation sequencing or whatever analysis we have there to actually get results from our tissue biopsy. And you can really see that there are 2 different timeframes. One is a relatively constant process. I think most pathology labs these days are really capable of having a very constant process with a very predictable turnaround time once the tissue enters the pathology laboratory. While actually the first part, actually getting to the tissue, is a very variable time frame, which can be very, very different. How long does it take to really schedule the appointment, have a patient come into a hospital, and really get access to the biopsy there? In contrast to this, when we now see how we actually get liquid biopsies, it's a much, much shortened process that we have there. Because at the end of the day, the only process that we have is to draw blood. We don't need to schedule a biopsy. We don't have to fix it. We really just draw blood, extract nucleic acid, and then give the nucleic acid for sequencing. And drawing blood is much, much easier, for example, to getting a tissue biopsy. I think many of us have done it at various laboratories. So this is really interesting because in addition to, the more insights into, longitudinal testing of liquid biopsy, I think we can also think about liquid biopsy of a different way of actually obtaining tissue or obtaining insights into the biology that we have there. And therefore, I think it's always very, very important when we think about the time that we need, for example, to get to a report. It's very important that we think really the time from a request to the final report and not just how long does it take, for example, to do a next generation sequencing, because I think this is where liquid biopsy can really be much, much quicker. And then while I don't show this here, I still want to remind everybody that also you always have to consider that not only the total duration, but also the time that there are actually hands-on things to do, or where we have certain machines that are actually taking their time for us, because this is also very important for the time that we need. And this is now very, very important because with this idea, we see the liquid biopsy can really give us much, much more insights. And I really briefly want to highlight two very interesting research conducted by my colleagues here at the MD Anderson, where they thought this really as a tool to reduce the turnaround time from liquid biopsies by also thinking about alternative eluates and not just blood. For example, this is a very, very interesting trial where they used fine needle aspirate supernatants, combined this with rapid molecular profiling to really significantly decrease the turnaround time compared to tissue-based testing. And this is really, really interesting because they saw that actually there was a very good correlation to the primary tissue, to liquid biopsy analysis, but the turnaround time was really significantly shortened by the simple fact that the access to the material that you're going to analyze, so the circulating nucleic acids that you have there, is much, much shorter when you now use this liquid biopsy approach as an alternative to the more classical tissue-based approach. And I will briefly only touch upon this because Dr. Ballister will also present during this webinar series today and give much, much more insights. But it's also very, very nice to see other alternative methods, for example, like cerebrospinal fluid, again, to be used as a method to decrease the turnaround time and increase really the access to your molecular report in a very short turnaround time. And this is very, very important when we now think about, we know the liquid biopsies are there. I think I showed you already some data that we can use it, but how can we now put it into new settings where we can use liquid biopsy, not to replace tissue-based testing, but to complement it and use it in a way that we haven't thought before to really reduce the time that we need to get a diagnosis for treatment stratification. And in line with this, I will end this presentation with a very important note on this. Because when we now think about access to oncology care, this is a very, very important study that came out last year, which really highlights across the United States the access not only to oncology clinical trial, but also highlight the access to oncologists. And what you can see is that there are really, really large parts within the United States where there are counties where there is no known oncologist present, really limiting access to care for the patients living in those places. And I think, and this is really just a thought that I want to end this presentation here, not as a recommendation to now use a liquid biopsy as a replacement for everything, but I still want to highlight, therefore, that in this regard, where it's really challenging to get access to this tissue, that liquid biopsy could really expand the access to precision oncology by the simple fact that the collection of blood is much easier compared to when we need a tissue-based biopsy. It can really happen in outside, less specialized centers compared to tissue-based testing, where we need much, much more equipment. And again, not as a recommendation to replace tissue biopsy with liquid biopsies for everything, but really as a thought when we think about liquid biopsy, that actually it is also a tool that can really help us to expand access just by the simple collection of material that we have there. And with this, I want to already come to the end of my presentation. I hope I could demonstrate to you that liquid biopsy of a minimal invasive approach that really help us to obtain real-time insights into tumor biology, and that we can actually use different sample sources that we have there. We usually most commonly use blood plasma, but I also showed you fine needle aspirates or cerebrospinal fluid, for example. I hope you now better understand that analyzing various sources such as circulating tumor DNA and circulating tumor RNA really increases the detection of actionable mutations, and is therefore a very, very interesting novel approach as an integrative liquid biopsy. We see very good correlation across liquid biopsy panels, and this really irrespective if you use a Hamilton or hybrid capture-based selection method, really highlighting the reliability of liquid biopsies. And then again, just to highlight that we can also use a liquid biopsy, not just for treatment stratification, but also to serially track changes during treatment and really to use it as a potential future method, to gain more insight on tumor intrinsic processes during treatment, and hopefully in the future as a tool to really change the treatment adjustment. And then lastly, I think it's very important that we all also consider liquid biopsy as a tool with a very rapid turnaround time that may expand access due to its easy sampling method, and can really therefore really help us to expand the access to precision oncology. And with this, I look very much forward to the question and answer session following by my presentation, and look forward to hearing your comments and questions regarding liquid biopsies. Thank you so much for your time.